Hello, everyone. Today I'm going to talk about the molecular phylogenetic relationship and morphological assessment of philodendron species in Central America. This project is in collaboration with Orlando Ortiz, Astrid de Mestier, Nils Köster, and Thomas Borschr. This presentation is organized in the next outline. First of all, I'm going to talk about the characteristic of the genus, background, my thesis topic, field and lab work, and then the preliminary results. Uh, about the characteristic of the genus, is the second large genus in the family Araceae, about more than 1,000 species, consists in hermi or epiphytes, nomadic vines, terrestrial or rhiophyte plants, is one of the genera with highest diversity in terms of morphology, and one of the diversity centers is in the tropical Andes in this region, and then there is another diversity center in the Talamanca mountain range between Costa Rica and Panama. About the inflorescence and flower morphology, Philodendron is one of the genera with spadic that have unisexual flowers, which means that here in this part, the female flowers are present, and this is the female flowers. And then in the center are present a sterile zone. This is the flowers from this region. And then from the end of the sterile region to the apex, the male flowers are present. And this is the male flower, and this is the state. About the leaf morphology, as I mentioned before, this is one of the highest diversity genus in terms of morphology, and this is one of the common blade shape that you can find in the nature, from narrow leaf, subcordate leaf, trilobulate, trifoliolate, pedatic composed, and pinnatifid blades. About the profile in the most of the species in the family Araceae, there are two structures that in the development genome stem that consist in the profile, cataphyll, and the photosynthetic blade. But in this case of the genus Philodendron, we have only one structure that we call it the profile, and then the photosynthetic blade. In this case, this is the common profile in this genus. This is the persistent profile, and this is the one of the profile that's become gelatinous and then fall down by fragments. This is another group that start rolling from the apex to the base and then fall down like this. And the last one consists in the marcescent profile and then becomes like fibers. About the infragenetic classification, there are three subgenera. The subgenus Meconostigma that consists in species from the Amazonia basin with about 21 species that consist in strong philodendrons, and the most of the species are terrestrial. And then the subgenus Pteromycon with distribution in Central South America and some Caribbean islands, and consists about 82 species. The most of the species are vines. And then the subgenus Philodendron with more than 460 species with the same distribution than Pteromycon, and the life form are variable. Regarding the background in the genus Philodendron, there are two recently published papers about the relationship between the three subgenera and the species in the subgenus Philodendron by Vasconcelos et al. In the 2018, but a few months later by Canal et al. In the same year. But the most of the species that they included are from South American distribution, and only a few species from Central America. And for now, we don't know how is the relation between among the species from Central America, from the Talamanca mountain range, east of Panama and the south of Mexico, with the South American species that they included in this paper. And only one article about the biogeography in the genome, but they also included a few species from Central America. For this reason, 2.5 years ago, I have the opportunity to start with my PhD with this topic, diversity of the genus Philodendron in Central America. evolutionary origin and diversity and species delimitation. With this possible objective, further complete the backbone phylogeny of the genus Philodendron toward an infragenetic classification. And today I'm going to present the preliminary results in this topic, and then the character evolution in Philodendron, and the last one, the species delimitation and taxonomic treatment of the Central American Philodendron Wilburi complex. About the field and lab work, we did field work 2 years ago in Mexico, Panama, and Costa Rica, and also in Colombia, recollecting as much as possible the species in the field. And then we split all of the plant to prepare the herbarium specimen, to prepare the DNA samples, and then we start with the first spirit collection with the three part of the inflorescence, the female stellar region and the male, and then we included cuts from the stem and the petiole for possible anatomy analysis. And also we are doing this documentation for the inflorescence in anthesis, taking picture from the front view of the inflorescence, lateral view, back view, and then we are doing this longitudinal cut in the spine to see how is the insertion of the spadic in the peduncle, and then to make a documentation of the shape of the morphology of the spadics, in this case to see how is the morphology of the female region, the stellar region, and the male, and also taking picture of the 3 kind of flowers in the spadics. For the DNA insulation, in the beginning I was using CETAP, but now we are using this kit, and we are having significant values in the quality of the DNA, which means with less contamination. And for the phylogenetic analysis, we are using this program, 5D, to edit for the addition of the ferrograms to assemble the sequence to generate the consensus sequence for these 3 markers. And then the maximum likelihood and Bayesian inference to perform that analysis, we are using the Corta server from the Freie University. About the preliminary results, the field work in Costa Rica and Panama. In Central America, there are 132 species, 17 in Costa Rica and 107 in Panama. And we collected in Costa Rica 81% of the species, and in Panama 85% of the species. Respecting to the lab work, in the last article, in the last paper by Canal, they included 224 accessions representing 173 taxa. Right now we are going to include 302 more accessions representing 132 taxas. Combined with Canal, we have 527 accessions. And this is the first phylogenetic tree with Central American species with these 3 markers. This preliminary phylogenetic tree consists in 336 accessions represented 240 taxas. Here there is the outgroup, and the light green color represents the subgenus Meconostigma, and then the dark green color represents the subgenus Pteromission. Both subgenus like sister of the whole diversity subgenus Philodendron. The species in the red color represent South American species, and then the blue ones represent the Central American species. Next. This is with zoom in the phylogeny to show you the relation. Here, this is the Adelonema genus that consists of a neotropical genus, and the Adelonema with the Omalomena, that is an ancient genus, is the sister group of the whole genus Philodendron. And then this is the subgenus Meconostigma species that consists in the most of the case terrestrial species, only 1 or 2 mypified species. And then the pteromission, that is completely different to meconostigma, that consists in vines, are together being the sister group of the subgenus Philodendron. Next, please. And here we have the first aggregation of clades, in this case, that consists in a species from the Brazil Mata Atlántica, Guiana Shield, Amazonas, and the north of the Andes, and a few species from the Caribbean island. The next, please. And here we have this group that consists in one of the clades with biogeography migration patterns with species from the North Amazonas, Chocó, and the first Central American clade with the species from the east of Panama, and then from species from the north of the Talamancan range, with some species with distribution from the Chocó to Central American in Costa Rica, Panama, Costa Rica, and Nicaragua, that consists in Philodendron sagittifolium and Philodendron tripartito. And then we have this second group with biogeography migration patterns that consist in species from Amazonas, Andes foothills, and then Amazonas, Andes foothill, and Chocó. And here we have a species from the east of Panama, from the north of the Talamanca, and from the south of the Talamanca. And some of these species here are endemic from Costa Rica or Panama. And then we have an endemic clade from the Chocó. Next. And as a sister group of the second Central American clade, which is species from South of Mexico, Central American in Costa Rica and Panama. And then we saw wide distribution species from Chocó to Costa Rica and Panama as Philodendron platypetiolatum and sulcicauli, and one species from the south of the biogeography Chocó that consists in Philodendron bonifaci. About the divergent time estimation, we have here that the subgenus Philodendron, the diversification time is between 35 to 45 million years ago, and here we have the Central American clade that consists in a group that in this preliminary result is between 18 to 25 million years ago. And this is only to show you that the species for each clade are consist or belong to the same biogeography area. In this case, we have the South American taxa that consists in the green line here, and then Caribbean taxa, the red one, and then the Central America taxa consists in the blue one. And here we have this species that consists in the widespread species like Philodendron ederaceum, Jacquini, and Fragantissimum. But this sample here consists in the accession from Central America, Costa Rica, or Panama. But here we have one species endemic from Mexico and one species endemic from Guatemala that are in the South American clade with the species from the Guiana Shield and from a species from the Caribbean island. And it should be a probably migration pattern from the Guiana Shield to the Caribbean island to Mexico. And then the two Central American clade, most of the species are from this area in Costa Rica and Panama. About the preliminary morphological assessment, we are using this clade to evaluate the morphology of the inflorescence, because this is one of the structure that loss of the characteristic in the herbarium samples. We are taking pictures for all of the stages of this inflorescence in different parts, like I mentioned before, looking for some characteristics that we can use to explain the relation between the species in the clades. And we found in this clade that these species are related in the morphology of the spathe and also in the morphology of the spadix. You can see here that the sterile region are like this kind of ring, and is present in, this is only 3 species, but we evaluate 6 or 7 species, and these characteristics are present. In the case of the flower, it's useful, this characteristic, because it depends of the species, the amount of locules or ovules could be different. About the blade morphology, we are doing exploration, but this is all of the species from this clade. You can see how variable is the blade shape here. And we are looking some patterns in the venation because we know that the blade shape is not constantly, but maybe the venation should be an important characteristic that we can use for this clade. What is next? We are exploring another region or markets to get more resolution because in some part of our preliminary result, we don't have a well resolved the branch, and we are looking for some region that we can get more information. In this case, one of the recommendations was RPS4. And then we are working in the morphological database, but also we want to include anatomical structure of the stem and the petiole, and also the chemical profile of the leaf. If we obtain founds, we are available to make phylogenomics approach, but it depends if we got the money. And then if it is possible, we are going to use 3 or 4 species for each case to make the exploration. And thank you very much for paying attention. It was a pleasure for me to be here. Thank you.
Thank you, Marco. Any questions?
About this species that were widespread and you found in the iguanas. So any idea how they were able to travel over the whole the Caribbean?
No, but there is only a few samples, but it's not representative from the whole Caribbean island. It's only in the Trinidad and Tobago Islands and Guadalupe. We need to include more species to see these patterns of migration around the Caribbean.
Thank you. Very nice talk. Thank you.
Anyone else? Okay, thank you, Marco.